Hello everyone, good afternoon. I'm Rodolphe Clerval. I'm the CEO of Coave Therapeutics. So Coave is a genetic medicine company, primarily focused on the ophthalmology and CNS field. The company is based on a unique, actually, delivery technology called ALIGATER, and I will come back on this one. It's an orthogonal platform in the viral space where we basically conjugate ligands on surface AAV to improve their efficacy and safety. From this platform, we are developing our internal pipeline of product with a lead program to enter IND-enabling study next year and being in clinic in early 27. On the top of internal developments, we have multiple ongoing collaborations with pharma and biotech company, evaluating currently our technology for a potential target that will not develop internally. We have raised 32 million Series A earlier this year. The round was led by Novo and BPI, and the company is around 30 people today. We're based in Paris, France. So what is our platform? ALIGATER is for AAV ligand conjugates. So we are inspired from antibody drug conjugate or conjugation of other entities where we have found a unique chemistry, which is not the classical click or maleimide chemistry. We are using a different chemistry, one-step chemistry, which enables to conjugate different ligands at the surface of AAV, and we demonstrated this technology works on LNP as well. So what we do, basically, we decorate those AAVs with those ligands, which change dramatically the biodistribution, the tissue specificity, but also the safety of the product. And the platform is very versatile. We could potentially address multiple kinds of tissue, but for the time being, we are primarily focusing on the eye, the brain, and muscle. So what is unique in our technology is we are not modifying AAV prior conjugation. So we can start with an existing AAV, and we have done that on all existing serotype. And we first design a ligand to address a certain receptor of mechanism of action, so which enabled to design a ligand for human biology. And we screen the ligand first. When we have a family of ligands that demonstrated a right interaction with the receptors, we conjugate on different serotypes. So it's much efficient and faster way to design a new capsid as opposed to what our competitors are doing, and I will come back to this one. So it's the other way around, actually. Plus, we can play with different level of ligands at the surface of AAV to play with the avidity of the capsid. The competition in the field, and you have here different names on the slide, are using genetic engineering on mostly directed evolution to generate new variants. So the way they do that is they create billions of different variants. They screen intensively on multiple animal studies, and they have to go to large animal. Then they find a variants that have a certain property. Without the certainty, this property will actually translate in human because these variants have been selected on certain receptors that could vary between a primate and a human. So this is the big difference compared to what we do, where we design these ligands on specific human receptors. In vitro, we screen it, and then we apply it to the capsid. We have actually two main family of conjugated AAV. AAV conjugated with sugar-based ligands, where basically here the role of those ligands is to interfere between the AAV and its natural receptors, especially the one presented in the extracellular matrix. And it actually improves the biodistribution and the spreading of the vectors when those vectors are injected in the tissue or in minimally invasive route of administrations. And using those sugar-based ligands, we are focusing on two main programs, one for targeting the brain through the intra-CSF route, and one targeting the retina through the suprachoroidal route of administrations. Besides, we have another family of ligands which are based on peptide and cyclic peptide. Those ones are specifically designed to tissue targeting. So we have designed the ligand to redirect an AAV injected in the IV route to, for instance, crossing the BBB or targeting muscle. Talking about suprachoroidal capsid, one of our lead capsid, why suprachoroidal route? This is one of the 2 routes that could be performed in office by the ophthalmologist because it doesn't require the surgical procedure. It has a lot of implications. This is a procedure that could be used to treat prevalent disorders, and it's the only route that could that enabled to target the back of the eye, and especially the RPE on photoreceptors. The current other route, which is IVT, has proven limited actually efficacy because while the route is easy to be performed when you inject in IVT, most of the capsid will not go to the retina but actually transduce the anterior segment of the eye, which lead to inflammation. So we think that improving gene therapy or developing new generation of gene therapy for large indications will require to develop the best vector for the suprachoroidal route. And today, companies developing on having clinical stage programs in gene therapy are using traditional AVs, AV2 and AV8, which have not been optimized for this route. So we see that those vectors are able to cross the choroid and go to RP, but have a very limited transductions of photoreceptors and have a very limited spreading. We are developing a capsid which goes through the whole retina layers and spread across from the periphery to the macula. So this is one of our studies we have conducted in NHP, where we demonstrated that our capsid, which is a conjugated AV2, has 11-fold better transductions of the retina and 21-fold better transductions of the RPE choroid. What you have on the bottom right is actually the spreading. We see that this capsid, when injected in the periphery, can spread to the macula while other capsid doesn't spread. So it makes this capsid as a best in class and probably first in class for this route of administrations. We have a second NHP study that have been completed recently, which have even better results than the one I'm presenting today, but will be presented later in the year in Congress. For the CSF route of administrations, today we are also another conjugated AV2 capsid using, again, sugar ligands. We compare it to AV9 in a head-to-head comparison in 2 studies. The first one here was in primate, where we demonstrated that our conjugated AV2 has actually 10,000 from 100 to 10,000-fold better transductions compared to AV9, including in deep structure of the brain, while this capsid is detargeted from the dorsal root ganglion, which is one of the main limitations with AV9, where we have a massive transduction of GRG leading to actually potential peripheral nerve toxicity. In IV administrations, we have 2 programs with 2 different targets. So we have actually developed ligands that interact with the human transferrin neuron receptors, which is a receptors well known and characterized for crossing the BBB. There's multiple clinical stage programs that are using these receptors with a specific shuttle to cross it. There's actually even a product approved in Japan that are using TFR1 to cross the BBB. And we have another set of peptides that are designed to interact with integrin, which are involved in actually fibrotic tissue and muscle. So this is one of example of what we are doing with TFR1. With our technology, we can play with different AV serotype, plus serotype that we have designed to detarget them from tissue. So we are the only technology where we can take an AV, make it totally inactive, and redirect it to a specific target. And what you have here is a demonstration with 2 different peptides, 2 different affinity. We can actually increase the level of conjugations on AV9, and we see that this receptor, the more ligand we put, the more interaction we have the receptors, and we can play with also we replicate the affinity of the receptors. So when we use the receptors with low affinity, actually we have a better the conjugated capsid has better interaction with the receptors. How that translates in vitro, what you have on the slide, on the left of the slide, is actually 2 cell line, one expressing the human transferrin receptor one and one not expressing it, and you see that the conjugated AV9 actually transducing through the TFR2, while AV9 is not transducing or has limited transduction in both cell type. In the middle figures, you have actually demonstration that the more a ligands we put at the surface, the more transductions of the cells we achieved. And on the right, it's a competitive assay where when we put those conjugated AV9 and we transduce cells in presence of actually human transfer in one ligands that compete with the AV, we actually stop or we reduce dramatically the transductions. And if we use a conjugated AV9 with the same construct but without the binding moiety, there is no transduction. So this study demonstrated really that we have developed technology that enable a cell and even receptor-specific transductions of AAV by conjugating the right ligand. Another example is actually using another peptide called RGD. This peptide binds to integrin receptors. Here we have conjugated different AV, AV9, AV9KO, AV2KO, which are partially targeted vectors. And again, when we conjugate and increase the conjugation level, we have an increased transductions. And what you have on the right, it's another integrin called beta-6, where we compare a conjugated AV9 on cell line expressing or not expressing the beta-6 integrin. So you see that the transductions of our conjugated AV9, which are the blue bar, is really specific from on the cells that express the beta-6, and they have been compared to two benchmark AAVs that have an RGD peptide integrated in the capsid, one from Geneton and one from another institutions. And we demonstrate clearly that our conjugated AAVs are much efficient than those one and have more specific to beta-6 integrin. So this is for the capsid technology. So our strategy is to develop product, but we think a successful product will start with a successful vector and delivery technology. So we have defined a target vector profile for each of our vector programs that need to be fulfilled before combining this vector with a specific payload. Our strategy will be to address a validated target first with our vectors, so developing best-in-class programs. And for each of our capsid that you have here, it's a suprachoroidal, IV for the muscle, CSF, and IV for the brain. We have pre-identified targets that we are already construct in place, and we are in discovery stage. One of the most advanced programs is using the suprachoroidal capsid for a wet AMD, second generation wet AMD program to be developed. And upon the validations with those validated targets, we can apply this technology to other targets which are less de-risked. So this is it for the presentation. Thank you for your time, and happy to take questions if any.